Welcome, Duke alumni and friends, to the session of the Duke Alumni Forever Learning Institute, which is an interdisciplinary educational program organized in a set of thematic courses. I am Squire Servance, your host for this evening. I am the managing partner at Servance Bio, which is an impact-minded life sciences focused investment firm. I am also on the board of the Duke Alumni Association, so I'm excited to be here tonight. Thank you for joining our program, Thinking About Brains, Masterminding Prevention and Repair, which is co-sponsored by the Duke Institute for Brain Sciences. This program is part of our theme, Fortifying the Body and Brain, which is inspired by the Duke Science and Technology Initiative. Moderating our discussion tonight will be Professor Nicole Schramm-Sapyta, who's Associate Professor of the Practice and Associate Director for the Duke Institute for Brain Sciences. Welcome to the Forever Learning Institute. I will now turn it over to Nicole.
Hi, thank you so much, Squire, for that kind introduction. And thanks to all of you from all around the country for tuning in tonight. It's been great to see all the locations coming in across the chat. So tonight, I am so excited to introduce three of our amazing Duke researchers to speak with you. And they're really bringing really fresh perspectives on neurodegenerative disease to the world of science and the world of medical practice. So just to put us all on the same page, neurodegenerative disease is the broad category of diseases in which our brains, and specifically the neurons in our brains, break down and deteriorate over time. You're probably familiar with names like Parkinson's, Alzheimer's, amyotrophic lateral sclerosis, or ALS, which used to be called Lou Gehrig's disease. Many other forms of dementia fall under this category. All these illnesses fall under the category of neurodegeneration. And tonight's speakers are focusing on different aspects of these illnesses, from the very subcellular level to the whole brain, to the family, and to the things that we can do to impact the quality of life of those impacted by these different forms of neurodegeneration. So I hope you are as excited about their talks as I am. I've just had a sneak preview, so you're in for a treat. So our first speaker tonight will be Dr. Chantell Evans. She's an assistant professor in the Department of Cell Biology, and she's new to Duke, having been here for just 1 year. She'll talk about something that I've been curious about since I read the book A Wrinkle in Time in middle school, which is mitochondria. And what role do mitochondria play in neurodegeneration? After Dr. Evans, our second speaker will be Dr. Rick Bedlack. He's a professor and clinician in the Department of Neurology, and he spent his long career here at Duke finding a wide range of ways to improve the quality of life and to find novel treatments for these difficult diseases. And finally, our third speaker will be another new professor to Duke, Dr. Cameron McIntyre. He's a professor in the departments of Biomedical Engineering and Neurosurgery, and his talk will describe his work to bring technological advances such as deep brain stimulation and virtual reality to develop better treatments for these brain ailments. So thank you all for coming, and I will now hand off to Dr. Evans.
Thank you very much for that kind introduction. So today I'm going to tell you guys a little bit about the work that my lab is doing, which is investigating mitochondrial quality control dynamics in homeostasis and disease. So neurons are highly polarized, terminally differentiated cells, meaning that we must maintain our neurons for the lifetime of an animal. And our longest neuron extends from the small of your back all the way to the tip of your toe. And so if you think about that long distance, there are molecules and proteins and organelles that need to traffic along this long axon in order to get things out to where they need to be. And so this spatial temporal signaling is very important for neurons for supplying both proteins and organelles. And neurons also rely heavily on mitochondria to support local energy demands at fusion sites that can be located far away. So to put this into a better perspective, what I'm showing you here is a motor neuron drawn to scale. This tiny red dot right here represents this soma, and then this long continuous line winding back and forth is the axon. And so I think from this perspective, it begins to show how important it is to know where a protein or organelle may be within this long axon, and how maintenance mechanisms that are defective over time can lead to the progressive functional and structural degeneration of the neuron. Now, mitochondria, I always like to take it back to high school biology, where we learned that mitochondria are the powerhouse of the cell. And so they are highly dynamic organelles that are important for maintaining cellular energy through ATP production. But we now know that they do a lot more than that. They're also involved in cellular metabolism. They can trigger the death of the cell, and that they're also important for calcium storage. And so more broadly, my lab is really interested in understanding how neurons are supported and maintain a healthy mitochondrial network in these spatially extended networks for the lifetime of an animal. If we go back to this and look again at this extended neuron and axon, we can see that mitochondria are located throughout. Here I'm just showing you the cell body so you can see what the mitochondria look like. This is a confocal image. I like to think of it as a bowl full of spaghetti. There are individual noodles within that bowl, but it's very difficult to see where one begins and ends because of how dynamic these organelles are. And within a single neuron of the substantia nigra, there are about 2 million mitochondria. And we know that there are millions of neurons within the brain. So you can do the math to understand how many mitochondria are located throughout the brain. And so if we look, we can see that over time, as we just age normally, that there's increased mitochondrial stress as well as dysfunction. So what I'm showing you here is a brain region of a rat in this young 10-month mouse. You can see these nice, healthy mitochondria. However, if you look at a 22-month-old mouse, rat, which is nearing the end of its life, you see that these mitochondria now look swollen and starting to look bad and not functioning as well as they should be. And so we're interested in mechanistically defining these various quality control mechanisms within the neuron to understand how they functionally cooperate during homeostasis and disease. It's now thought that there are multiple quality control mechanisms that work together to maintain mitochondrial health. And today I want to talk a little bit more about this mitophagy pathway. So mitophagy is a pathway where neurons clear or remove damaged mitochondria from the cell. There are multiple forms of this mitophagy pathway, but what I'm showing you here is just a schematic of what happens. So typically you'll have some type of damage that happens to your mitochondria. And then this triggers a cascade where you get particular proteins getting recruited to this damaged mitochondria. Eventually it gets engulfed into an autophagosome and then confused with a lysosome where lysosomal enzymes will start to degrade and break down and remove that mitochondria from the cell. And so this pathway allows for, again, specific removal of just damaged mitochondria. And so what I've always found interesting about studying mitochondria in this mitophagy pathway are their implications to neurodegenerative diseases. So a hallmark of many neurodegenerative diseases is this accumulation of dysfunctional mitochondria. And we've seen that mitochondria dysfunction is implicated in Parkinson's disease, Alzheimer's disease, Huntington's disease, as well as ALS. So these are just implications, but there's also genetic links to these. So if we look with these Parkinson's disease-associated mutations, we find two proteins, pink1 and parkin, that are in this mitophagy pathway that are mutated in both sporadic and familial cases. We also find that two proteins, TBK1 and optineurin, which are involved in later stages of this mitophagy pathway, are implicated in ALS, and both, again, sporadic and familial cases. But what I find most interesting is a statistic that I found recently that says by 2040, neurodegenerative diseases are predicted to overtake cancer as the second leading cause of death worldwide. And so because of this, I think it's really important that we start looking now for finding biomarkers or treatments or ways in which we can cure these neurodegenerative diseases. So one thing that we can do within our lab is we can take these previously found mutations that are associated with these various diseases and ask what effect do they have on this mitophagy pathway. So what I'm showing you here is a Parkinson's disease-associated mutant of this one protein, parkin. And then we can look at what effect it's having in the cell. So if we damage our mitochondria within the cell, we can see that under wild type conditions, this Parkin protein gets recruited to these damaged organelles. It recruits the second protein, optineurin, and then you'll get engulfment within an autophagosome and then removal of these damaged organelles. However, if you're expressing this Parkinson's disease-associated mutant of Parkin, while it still gets recruited to the damaged organelle, however, it doesn't recruit optineurin. So we're stalling this mitophagy pathway, and therefore these damaged organelles are just sitting and remaining within the cell. We then also looked at longer time points, and we can see the exact same thing. So this suggested to us that mitochondrial turnover may be a key mechanism contributing to neurodegenerative diseases. So you can imagine in a healthy neuron, this mitophagy pathway is happening, it's removing these damaged organelles. However, if you have neurons that are expressing any of these disease-linked mutations, this is either going to slow or stall this mitophagy pathway and lead to a buildup of these damaged mitochondria, which could lead to a decreased neuron health and then eventually cause degeneration. So this suggested to us that the time course for the removal of these damaged mitochondria may be very important for these vulnerable neurons. So what we decided to do was see if we could, in primary hippocampal cells, look at how quickly some of these proteins are getting recruited to the damaged mitochondria. So within an hour of damage, we were able to see the recruitment of this upstream protein Parkin. Again, it co-localizes and forms these rings around the damaged mitochondria. We also see ubiquitin, TBK1, as well as LC3. And so all of these occurred within the predicted timeframes of what was previously characterized in other cell types. So again, we can see within an hour of mitochondrial damage, we get engulfment of these damaged organelles into an autophagosome. We then wanted to look downstream to see what would happen, and do we see full degradation and removal of these damaged organelles within the cell. And when we did this, we were really surprised to see that these damaged organelles are not getting as removed as quickly as we would suspect. So we did this cool pulse chase experiment where we could look at different mitochondrial populations based on age. We would damage our mitochondria, and then we would go back and look a full 24 hours later to see had these damaged organelles been removed from the cell. So when we did this, again, we can see our optineurin ring, and if we look inside, we can see that these old mitochondria that have been there for 24 hours are still not getting removed from the cell. What's more interesting is they're not even fused with the lysosome yet. And so we think that there's this efficient sequestration within an autophagosome, but there's this slow, inefficient degradation of engulfed mitochondria, suggesting that the removal of these damaged organelles occurs hours to even days after initial damage. And what's even more interesting is that we compared this to other cell types. We do not see this happening in other cell types. So we think it's something specific about neurons that make them more vulnerable. So to conclude, we've characterized this novel mitophagy pathway where we're looking at the turnover and dynamics of these damaged organelles. We see that turnover is very slow in neurons and occurs hours to even days after damage. And so we think these slow kinetics may enhance the neuron's susceptibility to disease-linked mutations, which could be leading to neurodegeneration. So just to conclude with thinking about some greater implications of this, we think it's the combination of these multiple factors of this slow mitophagy kinetics and expression of these disease-linked mutations that's leading to neurodegeneration. And now we're following up on these studies to ask questions about why is this mitochondrial degradation in neurons slow, and also is slow turnover rate exclusive to mitochondria, or is it an inherent property of neurons? We know that aggregated proteins accumulate in, and that's also a hallmark of many of these neurodegenerative diseases. So we're trying to see if they're that the removal of those aggregated proteins may also be associated with this and be leading to neurodegeneration. And so with that, I will conclude, acknowledge the members of my lab. We just had our 1-year lab anniversary, so that's us celebrating, as well as my funding sources. And with that, I will stop sharing and pass it off to Rick.
Thank you, Chantell. Great talk. All right. Well, thanks to the Forever Learning Institute for giving me this opportunity to tell you about some of the more unusual research I do. And I've titled this talk, Untangling the ALS X-Files. So just by way of background, people living with ALS, and I would imagine any disabling, life-shortening, currently incurable illness, often consider alternative and off-label treatments, things that I call AOTs, that they find on the internet. And the proponents, the people selling these kinds of products, make some extraordinary claims about them. ALS breakthrough, guaranteed to work, perfectly safe. Unfortunately, the information that's available to back up these claims is often incomplete and sometimes, frankly, inaccurate. And this can lead to an overestimation of potential benefits from these products and an underestimation of potential harms. And sadly, there have been harms that have come from the pursuit of AOTs, even ones claiming to be perfectly safe. There are financial harms. Some of these products cost hundreds, some cost thousands, some cost hundreds of thousands of dollars. There are physical harms. We've found evidence of infections and blood clots and tumors comprised of a donor's stem cells and even deaths at some of these clinics. And finally, there can be scientific harms. And this has to do with enrollment. It's very difficult to enroll patients in trials of rare diseases like ALS. And we have certainly seen that patients who would have been good candidates for trials opted out of participation, instead pursuing an AOT. So frustrated by all this, in 2009, I decided to do something about it. I started a program that I called ALS Untangled. I wanted to bring together a group of clinicians and scientists that would systematically assess these AOTs with the ultimate goal of helping people with ALS make more informed decisions about them. And my motto was going to be like the motto of my hero, Fox Mulder from the X-Files. The truth is out there, but I know it's not always easy to find. There's 3 parts to this program I invented. The first is inputs. Where do we get our ideas? The second is investigations. How do we actually investigate and ultimately review these different ideas? And finally, outputs. How do we get the information that we learned back to patients and families who are interested? Everything in this program revolves around a website, www.alsuntangled.com.org.net. So inputs are ideas that come from people living with ALS and their families, and they come through social media, they come through email, they come through face-to-face visits with one of the ALS Untangled team members. Once we get an idea, we put it on a virtual bulletin board, the future reviews section of our website. And we've gotten a lot of these ideas, over 500 now currently on this section of our website. So many that I've realized I probably won't live long enough to get to review all these ideas. And so we've had to prioritize them in some way. And we do it actually in 2 ways. First, we let everyone vote. So simply by clicking the vote button next to any idea, you will see that it moves up on our priority list. And second, by giving it a multiplier. And the multiplier has to do with how much useful disclosable information we can find. And the reason we need that is even though some things might have a lot of votes, if any question that we ask the person who owns it results in a threat of a lawsuit, we're probably not going to be able to review that. So it gets a multiplier of 0. Gets a multiplier of 1 for almost any other circumstance, except if there's actually a published clinical trial or case series on this idea in people living with ALS, we multiply the votes by 2. Just like Fox Mulder had a team, I'm blessed to have a team, and it's really gotten quite big. I now have 130 clinicians and scientists working on this with me from across 11 different countries. There never was, and to my knowledge still isn't, anything else like this in any other disease state. So we've kind of had to invent a way to do this, and it's evolved over the years. But we now have standard operating procedures that guide everything we do, from how we gather information to how we write, crowdsource, and ultimately publish these reviews. And all of our reviews revolve around something we created called the table of evidence. So we care about 5 categories for each idea. Mechanistic plausibility. What is this thing supposed to do? And does that make any sense with what we know about ALS? Preclinical models. Has this thing ever been put into any test tube, cell, or animal model of ALS? And if so, what happened? Case reports. What are people on the internet and social media saying that tried this thing? Trials. And finally, risks. What bad things could happen if somebody tries this, and how likely are those to happen? And within each category, we assign a letter grade ranging from A to F. Just like back in school, A's are what you want, F's are what you want to avoid. But to give you an idea of how this works, take, for example, the middle evidence category of case reports. If all I can find on the internet are people with ALS who say they tried this and had bad things happen, you're going to get an F in that category for that idea. If there's one person somewhere on the internet saying they tried that and it helped them, but I can't find them, I can't get their records, I can't prove they had ALS or had benefits, you get a D. If I can find that person, get their records, and independently confirm their diagnosis and improvements associated with the product, you get a C. If there's more than one person like that, you get a B. And if there's multiple people like that published in a peer-reviewed journal, you get an A. And so once we have a draft, we circulate it amongst the whole team. We give everybody 2 weeks to weigh in. We incorporate all those comments. That's called crowdsourcing. And then we submit it to a peer-reviewed journal. It gets one independent peer review. We respond to that. And ultimately, it gets published in the journal called ALS and Frontal Temporal Degeneration. It also gets published on the completed review section of our website. Every one of our reviews are published through something called free open access, meaning patients and families never have to pay a penny to read any of these. Also, if you look on that section of our website, in case you don't want to read the whole review, we've got a summary of all the different grades in the different categories of our table of evidence. We've had to start updating some of our older reviews. And so if you see a little asterisk next to the name of an idea, that means it's been updated. We've also learned that some people would prefer to listen to their information rather than read it. And so we've started a partnership with an NIH consortium called CREATE. And in this partnership, a person who has lived with ALS interviews me about each of these ideas. And so there's sort of a verbal summary of what the review says. And finally, most recently, we've partnered with colleagues in Spain and Italy. We've learned that people in other countries want this information, and English may not be their primary language. And so there are now Spanish and Italian translations of much of our website. It's a tremendous amount of work. There hasn't been a single day in the last 13 years that I haven't worked on this in some way, shape, or form. It's easy to ask, is it worth it? Well, I could tell you we've won multiple awards for education and advocacy. But more importantly, I can tell you that people are actually reading these reviews. And so the journal that we publish in keeps a list of its most downloaded reviews. We currently have 8 of the top 10 most downloaded reviews in the history of the journal. Some of our individual reviews have more than 40,000 downloads, and collectively, the whole series is approaching 300,000 downloads, and our podcasts already have over 20,000 listens. I can also tell you that I've learned some amazing things, some unexpected things running this program. The first has to do with the products. You know, like many people, I was very skeptical about these products, but it turns out many of them actually do warrant further investigation because they have plausible mechanisms, positive preclinical data, and even positive, though flawed, human trials. And I've got lists of some of the products here. Some of my favorites that I would like to see further studies on: acetyl L-carnitine, basis, curcumin, hyperimmune goat serum, methylcobalamin, L-serine, and TUDCA. I also learned something surprising about ALS progression. Although I knew for most of my career that people with ALS could progress in ways that were wildly different from each other, I did not know that ALS progression could be different in a person at different times during the course of the disease. I learned this because I started hearing about all these patients that had an improvement in their progression rate coincident with starting a product. And it made me scratch my head and wonder, what happens if we follow people in clinical trials on placebos? Do we see the same kind of thing? And it turns out that we do. So we looked at a big database of patients with ALS who had longitudinal outcomes measured in clinical trials. We focused on just those patients who were on placebos, and we saw traces like the one that you see here, where there's progression for about 300 days, a lot of progression between day 300 and 400, then a period of long stability where there's no progression, and then even a period at the end where they seem to be improving spontaneously. So what I learned from this is it's very difficult to look at one case and conclude anything. I also learned that there's a handful of people that I never imagined are out there. 57 people now that I believe have completely recovered from ALS. So these are people who've sent me their medical records, and those records do appear to meet criteria for an ALS diagnosis. Those patients progressed to where they were disabled, and then they made a dramatic, in some cases, complete recovery.
I'm listening to it.
Some of these people were wheelchair bound and now walk. Some of these people were ventilator dependent and now breathe. This is really not described in any textbook. And so I had to ask myself, what do I do with these extraordinary cases? Well, it turns out there's a precedent for studying people like this who are unexpectedly resistant to a disease, and then having those efforts lead to better treatments for everyone with the disease. This happened in HIV with a group of people called elite controllers. And so I now have 2 research programs underway to try and understand these ALS reversals and to make them happen more often. The first program is called STAR, Study of ALS Reversals. And there I'm collaborating with that CREATE consortium to do whole genome sequencing in these patients, with a group in Cambridge called ALS TDI to look at RNA sequencing and proteomics, with Duke Radiology to look at imaging, with the Duke Microbiome Center to examine the composition of the gut microbiome, with the CDC to look at environmental exposures, and with the NIH Neurobiobank to someday look at the pathology of these patients. And the other program is called ROAR. This is a program of small pilot trials of some of the products, the AOTs, that were associated with these dramatic ALS reversals. It wouldn't be the first time that patients actually discovered the benefits of a product when researchers had no idea it was going to turn out that way. The most famous example of such serendipitous drug discovery is Viagra. Because I'm looking for such a huge signal, not just a slight slowing in ALS progression, but a complete reversal, these ROAR pilot trials can look very different than what industry tends to offer. They can have very wide, not narrow, inclusion criteria. I don't need any in-person visits. I can teach people to measure these dramatic changes and do it all virtually, which reduces travel burdens. I don't need placebo controls. If someone who's wheelchair bound in 6 months on one of these products walks again, that's not going to be a placebo effect. And so these trials have been wildly popular. They've been some of the fastest enrolling trials in the history of ALS. And so just to conclude, I've shown you that when it comes to AOTs, the truth is out there, as they used to say on the X-Files, but it's not always easy to find. I created this program called ALS Untangled that systematically reviews these AOTs in hopes of helping patients and families to make more informed decisions. Our reviews are very popular, and therefore I believe they are succeeding in our goal. But what really surprised me is that this program's actually led me to candidates for clinical trials that I never would have expected. And it's led me to a group of patients, very small number, but dramatic ALS reversals that I think, if we can understand why those happen, we might be able to make that happen more often, as in the HIV controllers. So thanks for listening to me tonight. I'll turn it over to my colleague, Cameron.
Great. Thanks. That was really inspirational, Dr. Bedlack. Really impressive work. So I'm going to talk to you tonight about deep brain stimulation, which is a medical therapy that is commonly used to treat a wide range of different neurological disorders. I missed the memo about, which form of deep brain stimulation I was supposed to talk about. So instead of talking about Parkinson's disease and neurodegeneration, I'm going to tell you a little story about deep brain stimulation for depression, but I think you're going to like it anyway. So deep brain stimulation is a relatively widely used form of what we call clinical neuromodulation. So an implanted electrical stimulation device that is used to treat a abnormal brain circuit. And so pretty much all of these different disorders that are listed here on this slide are things that we've had some degree of clinical success treating with deep brain stimulation technology. And so kind of analogous to cardiac pacemaking technology, except we put the electrodes in the brain. And the vast majority of these, several 100,000 patients worldwide that have had these devices implanted in them have been for the treatment of Parkinson's disease, but there are also this wide range of other disorders of interest. And tonight I'm going to tell you a little bit about this technology and its application to the treatment of intractable depression. There have been long histories of trying to treat depression with electrical stimulation technologies, both invasive, like deep brain stimulation, or non-invasive, like transcranial magnetic stimulation, and with varying levels of success and many, many failures along the way. And what we've learned along that process is that we're probably trying to intervene into one of the most complex circuits that we've ever interacted with. And we know very little about the circuitry that actually underlies these pathological conditions. And so, from a scientific perspective, the field has decided that what we probably need to do is take actually 2 steps back and think about how can we actually interrogate these brain circuits in human patients and use that information to then move forward with better hypotheses, better understanding, and real neurophysiological guidance. And so this is a brand new clinical trial that I'm a scientific investigator on, where they're implanting deep brain stimulation electrodes, these green electrode demarked locations, which are things that we've known in the past that we can put electrodes in that part of the brain. You can treat depression with success. But then also implant some temporary, what we call stereo EEG leads, or stereo encephalography, electroencephalography leads, to record the brain activity in these patients for a few days after their surgery, and then they'll take those leads out. And at the same time that this new trial was being put together, I was working on an idea of what we call connectomic brain stimulation. So there are many great advances happening in the world of brain imaging and brain modeling. And that information was full of all kinds of very complex 3-dimensional data about how the different parts of the brain are connected to each other. And so that Indian guy there is Samir Seth, and he is the PI on this clinical trial of deep brain stimulation for depression. And he and I have been friends for a long time. And he and I were very interested in this intersection between these brain models that we could build about the connectivity of individual patients' brains and where he was going to put all these different electrodes in this new clinical trial. And so once you start thinking about having 10 different electrodes in a patient's brain, the 3D geometry of things becomes very, very complicated, very, very quickly. And that really is a perfect test case for what we call these holographic visualization tools. So augmented reality headsets or head-mounted displays were just becoming available to the public, and we had already been working on how to build these software applications so that they could work in the context of patient-specific brain imaging data, with the idea of helping neurosurgeons be able to visualize all of this data in one environment so that they could really customize their surgical plan to each individual subject. So in 2020, 2020, we started actually doing these what we call prospective surgical planning experiments with a team of neurosurgical users, where we could do these group-based interactive discussions. So the guy on the far left is Nader Paradian. He's the chair of neurosurgery at UT Southwestern. Samir Seth's professor of neurosurgery at Baylor, and myself there, working together to try to think about, like, how do we actually maximize the therapeutic value of where we're going to put our electrodes, as well as the scientific recording value of all these different pieces and how they're going to interact with each other, so that we could customize the therapy to the patient. So, but that kind of academic exercise is really I mean, it's very cool and innovative and quite exciting, but it's not an FDA approved technology. So we had to figure out, how do we get the tool or get our results from that scientific environment into a commercial FDA approved neurosurgical planning platform. So that was a real fun kind of engineering problem. And then that kind of traditional planning environment has to then be transmitted into the surgical robot that's actually going to deliver these, kind of electrodes into the individual patient's brain. So there's all kinds of really cool engineering that goes into all of this, and that's its own story if anyone wants to hear about it some other time. So then, this was the very first patient in this clinical trial where we're using, kind of surgical robotics integrated with, as we said before, these really, kind of cutting edge patient specific brain models to then, actually deploy these electrodes into, an individual patient. As you can see, date-wise, this was, basically weeks before the world blew up into a global pandemic and everything shut down. But the good news is that we did get that patient implanted before the pandemic started. And what we're looking at here is examples of their clinical rating scales in their depression. And each one of those lines is a different clinical outcome measure that's used to measure depression. High numbers are bad, low numbers are good. And you can see the yellow line is, kind of the days when we were doing this electrophysiological recording after the surgery. And then the pink was then the monitoring phase of clinical, kind of outcome analysis for the patient. You can see progressively gets better. The red and blue lines represent the point of what would be considered remission from the depression symptoms. And so you can see after a few weeks, they really show a great deal of improvement. And then the blue area is when there was a blinded discontinuation of the deep brain stimulation on the patient. And you can see a pretty dramatic rebound in their effects or in their symptoms, then subsequently turning the device back on and immediately them getting better in a blinded way. So very, exciting. We've seen these kinds of things before, but the real, kind of breakthrough that is going on in this particular project is that we're collecting a level of electrophysiology data that has never ever been collected on a human scale before. So this is terabytes of electrophysiological recordings directly in the human brain, measuring their electrophysiological signatures of, all kinds of depression-related network activity. And so I think this is really groundbreaking work, and really, I'm just so proud to be a part of it. But as you remember, this first patient in that study was implanted right before the pandemic started. And then the pandemic hit, and we couldn't do any of this kind of interactive stuff in person, at least for about 2 years after that. So we had to develop an engineering solution to allow us to continue to do this interactive surgical planning and visualization so that the trial could keep going and that we could keep, kind of pushing these advanced computer models of brain stimulation into real clinical practice. And so we developed an infrastructure to allow us to do this remote interactive collaboration. So you were seeing a video that was taken from my office on Duke's campus, but then simultaneously, you have to imagine that each one of those blue peep avatars is, the view of someone else who was also part of that collaborative team. So, Dr. Seth's living room in Houston, or Dr. Paradian's office in Dallas, Texas. And so, kind of this idea that we can interactively communicate with each other and visualize in that way. And so this is just a quick, kind of picture of like, Dr. Seth, kind of grabbing one of those deep brain stimulation electrodes and then interactively kind of placing it into the brain. And there are these very specific brain connections that we want to stimulate. And so his goal is to come up with the exact right surgical trajectory and placement of the stimulation electrode so that you will interact with and stimulate the right circuitry in the brain. And that's something that's very specific to each individual patient. And so here in just a second, he will, boom, get it exactly the right place. And you can then visualize that, see that, and now have confidence that we're going to interface with the brain networks that we really need and want to get the therapy and therapeutic effect that we feel like we're trying to achieve. So I'll stop with that and turn it back over to our hosts. And I think maybe we'll be able to take some questions.
Absolutely. Yeah. Thanks to all of you so much. They were just wonderful talks. And the questions have started to come into the chat. So I'll just start firing them off. The first one I think was for Dr. Evans. And I noticed this in your talk and I heard it as well. It seems like neurodegenerative diseases are on the rise in the world. Do we know why that is?
Yeah, so it has to do with the fact that as each generation keeps aging, they're starting to live longer and longer, right? So as each generation lives longer, and we know that a lot of these neurodegenerative diseases are related to aging. So you start seeing onset around 65, 70. We have more people that are in that age group, and we know that 65 and older is the largest age group that's growing in the world right now, specifically people aged 80 and up. So because we have more of them, which means we have more onset of neurodegeneration.
So it's really a critical mass thing.
It's a critical mass thing, something that we need to think about now in ways to best prevent as our populations continue to age.
Absolutely. All right. So next question is for Cameron. Someone specifically asked just how does this neurodegeneration work? What are you stimulating? Is it release of dopamine? Is it serotonin? Like, do we know specifically how it's working?
So different circuits for different disorders, and that's really been the advance in some of these brain imaging things have given us ideas of the connections in the brain that we think we want to stimulate. And then the fundamental purpose of electrical stimulation is actually to artificially activate those axons that then subsequently release, targeted neurotransmitters into those circuits. So for each different disorder, they're, deficient in a different, kind of transmitter. So, in Parkinson's disease, we might be trying to target a circuit that hopefully might improve the release and control of dopamine, whereas in the neuropsychiatric applications, we're probably, interfacing with the network to improve the, kind of release and distribution of serotonin. So, it's really a ubiquitous tool that you're trying to manipulate the circuit to regularize it or enhance the, kind of transmitters of interest.
So it really depends on where the stimulation goes as to what neurotransmitters are released, which neurons are activated.
Absolutely. And that's where the surgical planning and the surgical, sort of like placement of the electrode is so fundamentally important. And, a millimeter can make all the difference in success or failure.
Mm-hmm. Mm-hmm. I'll give you another opportunity to since you addressed depression, I wonder, are there key differences between treating depression with deep brain stimulation versus treating the neurodegenerative diseases? Because there probably is a limit to when the neurons are gone. Does that limit the effectiveness of deep brain stimulation?
Yeah. So in Parkinson's, actually, it doesn't appear that is the rate limiting step. So let's say the nigral neurons, and once they're all gone, and they're not coming back. But interestingly, you can still achieve some degree of therapeutic benefit even once those neurons are gone. And so that's why we believe that it's a bigger circuit than just the nigral loss that actually translates into the motor symptoms of Parkinson's. And so, we don't really understand, for sure everything. But there's still some residual network effects, even when, all those neurons of interest have died. So.
Super. Thank you. I wanted to ask Rick as well. So I just love the array of all the different things that you've tested. And one of them that struck me was light therapy, which seems so benign. And I noticed in the score chart, it had very low risk, but also very low efficacy. So is there a similar one of those similar AOTs that is similarly low risk, but also showing some benefit? Like, what's the least harmful thing someone could try?
Yeah, it's a great question. So there's several things, as you can imagine, taking care of now more than 4,000 people with ALS, many of whom don't qualify for industry-sponsored trials. I've always got to have a list of like the most promising things they can self-experiment with, empower them to try things that have plausible mechanisms and reasonable safety. And then they won't, be on their own on the internet without that guidance. So right now at the top of my own personal list, I would put a product called curcumin. And this is a molecule, it's been studied. There's over 15,000 scientific papers on different forms of curcumin. It's anti-inflammatory, it's antioxidant, it interferes with protein clumping, which we call aggregation. It also probably changes the composition of the gut microbiome, which is a hot topic in neurodegenerative disease right now. So lots of plausible mechanisms. It has some positive preclinical data from the ALS mouse model. It has positive, though highly flawed clinical trials from other countries. It seems to be very safe. We just finished one of our ROAR trials. We don't have the data to present yet, unfortunately, but we just finished having 50 people on a water-soluble form of curcumin for 6 months. And so fingers crossed that we saw some improvements in some of them.
All right, that's a little bit of something, a little bit of hope there. That's great. Chantell, another question that's come through for you. It's getting back to the basics. How do mitochondria age? And how and why do they age? Is that just something the person asks? Is it cumulative oxidative damage, or is it some other mechanism?
Yeah, so it's a little bit of both, right? So everything in your cells are going to age. All proteins are going to age. They're going to need to be turned over. A lot of that is due to some sort of oxidative damage to those various molecules. So the same thing happens with mitochondria. We have done some calculations to determine that the half-life of a single mitochondrion is about 15 days. So it sits there for a substantial period of time, but all of the oxidative stress that those organelles experience as they're trying to generate the ATP and regulate the calcium and do all of these other things leads to damage within those proteins. And so then those organelles do need to get turned over.
Super. The same person asked a good question, putting your talks together. Is there any possibility that mitochondrial degeneration is also involved in depression?
Actually, there is. Well, I just ran a study not too long ago that was talking about this mitophagy pathway and its implications in anxiety and depression. So we're starting to learn a lot more about what these organelles are doing, and we're finding that they're involved in quite a lot of things. So definitely look it up. There's definitely some connections there.
Oh, that's great. The brain is also interconnected. We just can't escape it. Cameron, someone asks for you, how stable is the effect of deep brain stimulation? Is it irreversible, or do you have to keep administering it over time?
Yeah, so these are permanently implanted systems. And so patients, once they're in, they leave them on and they stay on, effectively 24/7 for decades. So there are patients, that have had deep brain stimulation systems implanted for well over 10, 15, even 20 years. So the batteries run out and you have to get the IPG, the pulse generator, replaced. But therapy continues. And when they do discontinuation studies, there's still an effect even, after many, many years. So almost all that track record comes from Parkinson's disease, where, really the therapy got its start, and really that's the vast majority of patients. But we seem to see that same issue or same situation in all the other disorders. So it's not a cure; it's a treatment of the symptoms. The stimulation is the network stimulation is the treatment of the symptoms, but it's not actually curing or changing the underlying network problems.
And is the stimulator delivering its pulses at a constant rate, or is it responding to some input from the brain or the body?
Yeah. So in the old days, it was always only a constant tonic stimulation But now the newest devices are actually being designed and engineered to act as a closed loop system so that they're using electrophysiological recordings to then pull back into a feedback loop that then updates the stimulation based on the patient's, physiology. And we call this closed loop DBS or whatever. And that's brand new stuff, great engineering problems and really quite exciting for the future.
Let's see. The next question that came through might merge Dr. Bedlack's and Evan's perspectives. Are there any lifestyle changes or behaviors that we can do to prevent mitochondrial degeneration or in general to prevent the progression of these illnesses?
So I think a lot, and especially in terms of these neurodegenerative diseases, it isn't so much the problem that the mitochondria are aging. It's a problem that the mechanisms to remove them aren't doing what they should. And so you get a buildup of those damaged organelles. And so when they're supposed to get removed, we know that with age, those removal mechanisms start decreasing with time. And so I think it's kind of a cascade of many, many problems of where the mitochondria are starting to go bad, the mechanisms to remove them are going bad, how they get degraded start going bad. And so it's kind of an accumulation of all of that. There has been some work that was looking about caloric intake and autophagy, which is one of those ways that you remove those damaged organelles. So I think that would be a good place to start to maybe look at some type of lifestyle.
Yeah, so Nicole, ALS Untangled is more about ideas to try to treat ALS, not to prevent it from happening. But of course, as we're plowing through the literature that supports, some of these ideas, we can't help but come across studies suggesting that certain things might reduce the risk of getting neurodegenerative diseases like ALS. And, one thing that keeps coming up for me over and over again is consumption of antioxidants, things like flavonoids, things like vitamin E. Now, it doesn't mean you have to actually take supplements. It turns out, you can get a lot of these things just from eating a healthy diet with plenty of fruits and vegetables. But multiple studies now have suggested that diets that are rich in antioxidants are associated with a much lower risk of developing neurodegenerative disease.
And I'll add to that one of the ways that we induce mitochondrial damage within our system is by removing antioxidants. And that's enough to damage the organelle.
All comes together as always. So we've got time for one last question. So I'm going to open it up to all three of you in turn. What is the next frontier? What are you looking forward to in the next few years in your research, and what's got you excited? So I'll let you go in order. Whoever wants to go first.
I'll go first. I'm most excited about the fact that we're finally starting to find some of the causes of ALS. We know that about 10% of ALS is genetic. So 10% of cases have a disease, causing mutation. And there's now more than 30 genes that have been discovered that can cause that 10%. And there's trials underway to actually address those genetic defects with things like antisense oligonucleotides. And we're even starting to get some clues about the 90% of people who don't have a genetic mutation. There appear to be things in the environment. For example, there's a neurotoxin called beta-methylaminoalanine that comes from blue-green algae that appears to cause some cases of ALS. And there's a virus called human endogenous retrovirus K that may be able to cause some cases. And there's now trials underway directed at those potential causes of ALS. And, I've been happy to have a few products that slow the progression, but what I really hope to see before I retire is a way to actually reverse some form of ALS. And I think the most likely way that's going to happen is by attacking causes of the disease.
That's wonderful. Thank you so much for all your work. Chantell, what about you?
Yeah, so I think our approaches are very complementary. We love to think about things very mechanistically and trying to understand what is going on with this pathway. And so we take the approach that until you really understand what's happening at a molecular level, it's difficult to do some of these treatments to understand what they're specifically doing. So we're trying to right now really characterize a couple of these pathways. And we think that we've found some very specific rate limiting steps within these pathways that we think that may be druggable targets. And with this, we're going to be able to speed up how fast the cell gets rid of these damaged organelles, which we think could be potentially therapeutic benefit. So I love what Rick is doing, and I'm starting to think about all of these different ways of like, maybe what could we do to test some of these to see at a molecular level what they're actually doing within the cell.
Just let me know, Chantell. I've got a lot of patients who'll be happy to have these new opportunities to work together.
Yeah.
All right, Cameron, your turn.
Yeah, I consider myself more of an engineer than a scientist, even though science pays the bills. But what I'm really excited about for the future is, there's been these huge advances in human brain imaging, human brain modeling, and I'm really excited about how we're going to take that kind of technology, that kind of computational approach to patient-specific medicine, and then integrate it with the surgical robotics that is also a big revolution that has happened in the last few years, especially in neurosurgery. So I think when you bring those two worlds together, we're going to have the opportunity to deliver things on a precision level that just really has never been able to be done before. And I'm really excited about kind of helping to engineer those systems to come together. And that delivery could be for stimulation, which is what I care about. But more likely, actually, the bigger impact will be for the delivery of, alternative, kind of new advances in, whether it's gene therapy or any other type of, kind of intervention that you're going to need precision delivery of an agent into the brain. And so I'm really excited about that, for the future.
All right. Thanks to all of you so much. This was wonderful. I know there were a few questions that we still didn't get a chance to address, but there will be time for that. I'm going to hand back to Squire for the closing comments.
Perfect. Well, thank you to Nicole, and our wonderful speakers, Rick, Chantell, and Cameron, for your great presentations today. And to our audience, thank you for those engaging questions, for joining us. We hope that you enjoyed learning more about the exciting work that's being done at Duke University.